السلام عليكم ورحمة الله. I am assistant lecturer لمياء هادي. I will development of the central nervous five brain vesicles that show following the cerebral hemisphere and the lumen of the telencephalon. Now المنطل zone show bilateral ventral thickening and bilateral dorsal thickening. Bilateral ventral thickening that the basal plate forming the ventral motor horn, gray matters of the spinal cord, while the bilateral dorsal thickening of the mantle zone forming the alar plate forming the dorsal sensory horn, gray matters of the cord. The dorsal and the ventral thickening are separated by sulcus limitans, and dorsal and the ventral midline of the cord show roof and the floor plate respectively, containing crossing nerve fibers with no neuroplast. The thoracic two lumbar three segments of the cord show the intermediate autonomic horns located between to the sensory horn and the motor horn. Now, histological differentiated in the first nerve cells. The neuroplasts have a transit central dendrite process. These dendrites disappear during the mantle zone formations and forming a rounded polar cells. Later on, the neuroplasts become a bipolar neuroplast and acquiring the primitive axon and a dendrite forming the multipolar neuroplast. The axon of the neuron of the basal plate forming in the fourth week the motor ventral root of the spinal nerve, while the axon of the neurons in the alar plate ascending upward in the marginal zone. Now, histological differentiated of the glial cells, the glial plast cells forming the glial cells of the mantle zone and the marginal zone, and also derived from the neuroepithelial cells. Some of the glial cells, as the microglia, are divided from the adjacent mesenchymal tissue, and the neuroepithelial cells finally forming the ependymal lining cells of the central canal of the cord. Now, myelinations of the spinal nerves. Myelinations in the spinal peripheral nerves occur by the Schwann cells at the fourth month, while myelinations inside the spinal cord occur by the oligodendrocytes also from the fourth month till the first postnatal life. Myelinations of the spinal peripheral nerve occur by the Schwann cells, while the myelinations inside the spinal cord occur by the oligodendrocytes. Now, positional changes of the spinal cord at the third month, the spinal cord at the length of the vertebral canal. You can see this is the full all the length of the vertebral canal. At the birth, the cord is ascending to the level of the third lumbar vertebra due to the rapid growth of the vertebra and meninges in comparisons to the cord. Therefore, the spinal nerve descending obliquely to reach the intervertebral foramen. In adult, the cord ends at the level of the first lumbar. Now, the abnormalities of the spinal cord, there are abnormalities of the closure in the neural tube. It involves the meninges, vertebra, muscles, and the skin. These include the spina bifida, involved a defect in the vertebral arch. First, spina bifida occulta with a defect in the covered by hairy skin, usually with no involvement of the nervous tissue. It occurs in the lumbar sacral regions. Here you can see, with no involvement of the nervous tissue. Now, a second of the abnormalities of the spinal cord, the spina bifida cystica, involve herniations of the meninges. It's called the meningiostia, or maybe with the underlying nerve tissue. It's called the meningio-myelocele. It occurs mostly in the lumbar sacral regions and associated with a defect in the nervous tissue. You can see this is the defect in the nervous tissue. Now, a third Regis cases or myelo cases involves a flattened of the neural fold tissue. You can see flattened of the neural fold tissue. Now, the morphogenesis of the brain. The basal and the alar plate develop as in the spinal cord, both in the hindbrain and the midbrain, while in the forebrain the alar plate grow further while the basal plate regress. Now, development of the rhombencephalon, or is called the hindbrain. The rhombencephalon consists of the two parts, the metencephalon and the myelencephalon. Metencephalon anteriorly and the myelencephalon posteriorly. The boundary between these two portions is the market of the potentate flexures. الميتencephalon which later forming anteriorly pons and posteriorly the cerebellum, while المyelencephalon forming the المedulla oblongate, and the lumen of the rhombencephalon that the fourth ventricle. Now, المyelencephalon differs from the spinal cord in that its lateral wall are verted. The alar and the basal plate separated by the sulcus, limitans, then the alar and the basal plate separated by a sulcus limitans. The basal plate, similar to that of the spinal cord, containing the motor nuclei. These nuclei are divided into three groups. The middle, the somatic efferent group, and the intermediate, the special visceral efferent groups, and lateral general visceral efferent group. The alar plate containing three groups of the sensory relay nuclei. The lateral of these nuclei forming the somatic afferent sensory group received impulses from the ear and the surface of the head by way of the vestibular cochlear and the trigeminal nerve. Somatic afferent, while the intermediate of the alar plate is called the special visceral efferent group, receive impulses from the taste bud of the tongue and from the palate, oropharynx and epiglottis, while the middle alar plate is called the general visceral efferent group, receive interceptive informations from the gastrointestinal tract and the heart. Three groups. Also, the three groups of the basal plate. The roof of the myelencephalon consists of the single layers roof plate consists of the single layers of ependymal cells covered by visceral mesenchyme, the pia mater. This is the roof plate. Consists of the single layers of the ependymals and the covering by the visceral mesenchymal, the pia mater. These two compound single layers of the ependymals and the pia mater, known as the thylakoid. Because of the active proliferations of the visceral mesenchymal matter, a number of the sac-like invaginations project into the underlying ventricular cavity. This is these tuft-like invagination forming the choroid plexus. You can see this is the tuft-like invagination. Is called the choroid plexus, which produce cerebrospinal fluid. Now, metencephalon, similar to the myelencephalon, is characterized by basal and alar plate and differentiated into pons that pathway for nerve fibers between the spinal cord and the cerebral and the cerebellar cortex. The second part forming the metencephalon, the cerebellum, coordinations center for the postures and the movement. Now, basal plate, each basal plate of the metencephalon containing to the three groups of the motor neurons. First, the middle somatic efferent group, which give rise to the nucleus of the abducens nerve, and the intermediate, the special visceral efferent group containing nuclei of the trigeminal and the facial nerve, which innervate the muscular of the first and the second pharyngeal arch. The third, the general visceral efferent group, whose axon supply the submandibular and the sublingual gland. Now, marginal layers of the basal plate of the metencephalon expand as it make a bridge for a nerve fiber. This bridge connecting the cerebral cortex and cerebellar cortex with the spinal cord. These portions, the bridge for nerve fiber of the metencephalon, is known as the pons or the bridge. In addition to nerve fiber, the pons containing the ponetine nuclei, which originate in the alar plate of the metencephalon and the myelencephalon. Now, the alar plate of the metencephalon also containing three groups of the sensory nuclei: the lateral somatic efferent, which contains neurons of the trigeminal nerve and the small portions of the vestibular cochlear complex. Second, the intermediate, the special visceral efferent group, and the general visceral efferent group. Now, a second part of the metencephalon, the cerebellum. The dorsolateral part of the inverted alar plate band medially forming the bilateral rhombic limb. These lips approach nerve to each other below the midbrain than الcaudally. The rhombic lips are compressed cephalocaudally by البوتينيت flexures, forming the cerebellar plate. A cerebellar plate which has a median vermis and a bilateral hemisphere. Initially, the cerebellar plate contains the neuroepithelial and the mantle and the marginal zone as other part of the CNS. Later on, many cells migrate from the neuroepithelia to forming the external granular layers at the cerebellar surface. These layers proliferations and forming most of the cerebellar cortex. The deeper of the cerebellar nuclei as the dentide nucleus develops from the neuroepithelial in the later developmental stages. Development of the prosencephalon or forebrain consists of the telencephalon and diencephalon. Telencephalon, which forming the cerebral hemisphere, diencephalon, which forming the optic cup and the stalk, the pituitary gland, the thalamus and the hypothalamus and the epiphysis. Now, the first part, the diencephalon consists only of roof plate and the alar plate. Roof plate forming the corrodus plexus of the third ventricle and the pineal gland or the epiphysis, while the alar plate forming the thalamus and the hypothalamus include the mammillary body. Now, the pituitary gland or is called the hypophysis. The pituitary gland development from the two parts, الرادكيس pouch and the infundibulum. الرادكيس pouch forming an ectodermal outbucketing in the third week at the roof of the stomodial oral cavity. This is the oral cavity. Anteriorly to the buccopharyngeal membrane. This pouch loses connecting with the oral cavity at the second month, forming adenohypophysis. Radke's pouch forming the adenohypophysis, the anteriorly pituitary gland, and forming البارس intermediately. This is the pituitary gland. Radke's pouch forming the anteriorly lobe or adenohypophysis and البارس intermediate, while the posterior pituitary gland forming from the infundibulum. Infundibulum projections of the diencephalon. These projections forming the neurohypophysis or the posteriorly pituitary gland, the pars nervosa. Now, the craniopharyngioma is a tumor arising from remnant of the Radke's pouch. It lies in the sala Turkicia and producing hydrocephalus and pituitary dysfunctions. This is the tumor of the craniopharyngioma. Producing the hydrocephalus and pituitary dysfunctions. Now, the second part of the forebrain, the telencephalon. Telencephalon consists of the bilateral cerebral hemisphere and median the lamina terminals and the cerebral hemisphere that development at the fifth week. The cavity of the hemisphere that the lateral ventricle communicate with the lumen of the diencephalon through the interventricular foramina of Monro. The basal part of the hemisphere grows, forming the corpus stratum that bulges into the lateral ventricle. This is the corpus stratum, bulges into the lateral ventricle. The wall of the hemisphere forming the choroid plexus near the roof of the diencephalon containing no neuroplast cells. Similar the plexus is formed in the lateral ventricle. This is the choroid plexus of the lateral ventricle, and this is the choroid plexus of the third ventricle. The expanding hemisphere cover the lateral aspect of the diencephalon, mesencephalon, and the upper metencephalon. The continuous growth of the cerebral hemisphere in anteriorly dorsal and inferiorly directions, resulting in the formations of the frontal, temporal, occipital lobes, respectively. The expansions of the hemisphere over the corpus stratum is very slow. However, the area between the frontal and temporal lobes become depressed and is now the insula, which is covered by the hemisphere. Cells migrate from the neuroepithelia of the hemisphere that forming so called the pallium. This migration is directed toward the surface and thus forming the layers of the cerebral cortex. Now, الcranial defect include the first الhollow البريزنcephaly which is a loss of the midline structures of the face and the brain, resulting in the single lateral ventricle, a fused eye, and a single nasal cavity. السكند the المننجيوسيل المننجيو الانسيفالوسيل and المننجيو الهيدرو الانسيفالوسيل. All these are caused by defects of the ossifications of the skull, mostly at the squamous occipital bone. It may be associated with herniations of the meninges and or the brain tissue. And the brain tissue. This is the meningeocele, and this is the meningeo encephalocele, and this is the meningeo hydroencephalocele. الثird the الانcephaly, which is the failure of the closure of the interior part of the neural tube, leading to loss of the cranial valve and degenerations of the brain tissue. Now, for the cranial defect, the hydrocephalus. Hydrocephalus is the abnormal increase cerebrospinal fluid that causes by obstructions to the flow of cerebrospinal fluid. The high pressure of the cerebrospinal fluid lead to the expansions of the skull and the degenerations of the brain tissue. This is the hydrocephalus. Now, the cranial nerve by the fourth week of development, the nuclei of all 12 cranial nerve are present. All cranial nerve arise from the brain stems except the olfactory and the optic, and of these only oculomotor 3 arise outside the regions of the hindbrain. In the hindbrain, proliferations centers in the neuroepithelial establish 8 distinguished segments that the rhombomeres. These rhombomeres give rise to the motor nuclei of the cranial nerve, the fourth, the fifth, the sixth, the 9, the 8, the 11, and the 12 nerve. The motor neurons of the cranial nerve are within the brain stems, while the sensory ganglia of the CNS are developed outside of the brain stem as a spinal nerve. These ganglia arise from the ectodermal thickened plaque and from the neural crest. Not all cranial nerve contain both motor and sensory. Now, autonomic systems. Autonomic systems at the fifth week, the neural crest forming the bilateral sympathetic ganglia. Some of these neuroblast cells migrate forming pre-aortic ganglia and the organ plexus. Neurons in the brainstem and sacral regions of the spinal cord from the preganglionic parasympathetic fibers. Now, suprarenal gland development from the ectodermal portions and the mesoderm portions, the ectoderm portions which forming the medulla of the suprarenal gland, and the mesoderm portions forming the cortex. At fifth week, the mesoderm cells between the roots of the mesentery and the gonads forming the large acidophilic cells which forming the fetal cortex or the primitive cortex of the suprarenal gland. The second wave of the cells from this mesoderm forming the definitive cortex around the fetal cortex. After birth, the fetal cortex forming reticular zone of the cortex only, neural crest cells inside of the fetal cortex stage from the middle side formations cords or clusters the chromaffin medulla. Thank you.